Assalamu alaikum everyone. My name is zohaib and I am a student of BS 7th Microbiology from Kohat University of Science and Technology, Kohat. The topic of my presentation is epidemic outbreak of anthroponotic cutaneous leishmaniasis in Kohat district, Khyber Pakhtunkhwa. The contents of my presentation include introduction, epidemiology, objectives, materials and methods, results, conclusions, and at the end, references. The research paper I have taken is from Acta Tropica, and the reason for selecting this research paper is because this is the first molecular study of leishmaniasis in local population of Kohat and internally displaced people coming from war-affected zone. The number of leishmaniasis cases has increased in last few years during migration of IDPs from cutaneous leishmaniasis endemic areas. Let's start with the introduction. Cutaneous leishmaniasis is most widespread form of leishmaniasis caused by Leishmaniasis tropica and Leishmaniasis major that invade the host macrophagic cells causing skin lesions on exposed part of the body such as face, arms and legs. The disease is transmitted by the blood-sucking sandflies, phlebotomus, whose 700 species has been identified all over the world, and out of which 37 species have been identified in Pakistan. These sandflies have wide range of habitat from tropical rainforest to deserts. They have a wide range of hosts including humans, livestock, dogs, chickens, vertebrates, as well as some mammals. Now let's talk about the epidemiology. The disease is endemic in Asia, Europe, and South America, and WHO has announced it as one of the most serious disease of world due to ever increasing number of new cases each year. The 10 countries include Afghanistan, Algeria, Colombia, Brazil, Iran, Syria, Ethiopia, North Sudan, Costa Rica, and Peru. Together accounts for 70 to 75% of global estimated cutaneous leishmaniasis incidence. Anthroponotic cutaneous leishmaniasis caused by Leishmaniasis tropica is reported as an endemic in many districts of KPK province had been reported. The vectors like Phlebotomus pepatasi and Phlebotomus sargentii are widespread in the area of Banu Dera Ismail Khan, Tunk, Kohat, Lahore, Mansehra and Nowshera. The province hosts more than 2 million Afghan refugees. Regarding leishmaniasis, Kohat is one of the most important southern district of KPK province. However, no precise report about leishmaniasis in local is yet available. To analyze the prevalence of cutaneous leishmaniasis, this is one of the objectives of our study. And to identification of causative species and vectors that are sandflies in local as well as IDPs habituating rural areas of Kohat district of KP, Pakistan. These are some of the results. Patients with active cutaneous leishmaniasis lesions in Quahar district during 2015 and 2016. Now let's talk about the materials and methods. First up, if we talk about the geographical and political profile of Kohat district, Kohat is a medium-sized district of KP province, located at a distance of about 47 kilometers from Peshawar, the capital of KP. The topology of the districts is dominated by the mountain and hills. The climate of Kohat district is usually dry with extreme hot in summer, around 40 degrees, and extreme cold and dry in winters, that is about 6 degrees. The average annual rain is about 400 mm. A prospective study was conducted from April 2015 to May 2016 at different villages of Kohat. 13 villages were selected according to the data of Basic Health Unit located near village who indicated uh, persons exhibiting cutaneous leishmaniasis. All right, for the sampling, the total of 29,865 individuals, including local population of 26,250 and IDPs uh, residing in the study villages were surveyed. A total of 300 smears were prepared from active skin lesions of willing patients. All the patients in study were referred to nearest government hospitals for free treatment. This sample were transported in ice and kept at minus 20 degrees uh, and until the further use for DNA extraction. Now, the collection of sandflies. For studying sandflies, reference points were set within a 1.5 kilometer radius from houses and infected patients. Sandflies were collected by sticky paper and some CDC light traps set outside and inside of the houses. These sandflies were stored in 70 to 80% ethanol till further use for morphological identification and leishmania testing by PCR. All the sandflies were identified based on external and internal morphological characters of the head and abdominal terminalia, which were slide mounted um, in the fluid following dissections with the sterilized forceps and microneedles. Female sandflies were used for parasite detection. Then the collection of rodents. The rodents were captured by locally made steel traps, baited with bread, peanut butter, vegetable, and fruits placed in the fields, bushes, and rock holes, and near the houses of cutaneous leishmaniasis patients. Uh, the spleen, liver, skin were collected and stored at minus 20 degrees until further use. Then after the sampling of all the required materials, the first step was the microscopy. Slides were prepared, fixed in methanol, stained with Giemsa stain, and examined under the microscope at 10x, 40x, and 100x magnification for Leishmania amastigotes. For extraction and amplification of DNA, the DNA extraction from all type of samples, that are human, rodents, and sandflies, were carried out using DNA extraction kit. Uh, the initial denaturation at 94 degrees for 3 minutes, and each cycle with the denaturation at 92 degrees for 40 s, annealing at 54 degrees centigrade for 1 minute, and extension at 72 degrees for 1 minute. The final extension was done at 72 degrees for 5 minutes. Now let's talk about the results. After the PCR, DNA extraction, and sequencing, the results were somewhat like that. A total of 1,359 local population and 140 IDPs causes were recorded from these villages. Among them, 52.5% were male and 47.5% were female. An overall prevalence of 9.03% recorded, including 5.1% in local population and 3.86% in IDPs. Gender-wise prevalence rate was higher in male in both local population and IDPs. Regarding lesions location, the most prominent area was uncovered body surfaces like face, hands, and feet. Now, these tables basically show the percentage of overall comparing high prevalence recording IDPs. And the next one in the right side, this shows the overall high prevalence observing male and female in all the populations. Now, the cutaneous leishmaniasis infected individuals were living in non-cemented house. The roof of these houses were either mud made with straw and wood. Cutaneous leishmaniasis was mostly recorded from villages. So 60% of infected population had different type of domestic animals like dog, sheep, goats, donkey, or chicken in their houses or in near their houses. In this survey, it was recorded that only 18.03% infected people had only limited knowledge about the disease. Now let's talk about the conclusion. The results of the present study reveal that cutaneous leishmaniasis has emerged as a major health problem in rural and suburban locations of district cohort. Factors like environmental change, global warming, and migration of people and reservoir from endemic to non-endemic areas resulted in high prevalence rate. These are some of the references. Thank you so much for your attention.